Okay, good morning, everyone. Welcome to VFPG. Today we have a talk by Dr. Emily Mitchell from the University of Cambridge titled Using Ecology to Unlock the Secrets of Ediacaran Evolution. Next week, we'll hear from Professor Moayet. Let's see, from Auvergne University. Their talk is titled The Origin of the Earth and its Silicate and its Early Silicate Evolution. So join for that next week. I'm going to go ahead. I'm going to introduce Emily Forrest, and then Emily, you can unmute yourself and prepare, but I'll let you know, or I'll let you know when to go ahead. So Dr. Emily Mitchell is a paleoecologist with a focus on sessile benthic Ediacaran organisms. She leverages exceptional preservation of paleo communities and life position with ecological statistical modeling to consider how the ecosystem structure and stability has developed with the introduction of key animal traits such as mobility, biomineralization, and macropredation. Dr. Mitchell received a bachelor's and master's degree in physics from Imperial College London, an MPhil in mathematics from the University of York, master's in research in environmental biology from the University of St. Andrews, and her PhD from the University of Cambridge with Professor Nick Butterfield. Her thesis was titled Quantitative Approaches to Understanding Ediacaran Ecology. After she was a Henslow Research Fellow with the Department of Earth Sciences of the University of Cambridge, followed by an NERC Independent Research and Senior Research Fellow at the Department of Zoology at the University of Cambridge. Where currently at the University of Cambridge, she's Assistant Professor at the Department of Zoology, Curator of Invertebrates at the Museum of Zoology, and Co-Director of the Leverhulme Center of Life and the Universe. So thanks a bunch, Emily, for joining to be with us today, and we're really looking forward to hearing your presentation. So please go ahead and take it away. You are still muted. Okay, great.
Excellent. Thank you very much, Alex. It's really wonderful to be here and to be talking to you about the work that me and my group have been doing over the last few years. So, as we're very much aware, the Ediacaran time period is one of the most important ones in the history of life on Earth, because it's when we first see animals appear in the fossil record. So just prior to the Ediacaran is dominated by microbial ecosystems for 3 billion years or so, and after the Ediacaran is where we get the Cambrian and we see the major animal phyla first appear in the fossil record. And it was during the Ediacaran that we see some of the most important innovations for animals first appear. So most notably large body size and the first animals themselves, but also the first evidence of mobility, the first bilaterians, biomineralizers, and then right before the Cambrian, the first evidence of predation. So the macrofossils of the Ediacaran form three major assemblages: the Avalon, the White Sea, and the Nama assemblage. There are, of course, many other sites, such as the Chibatan and sites in southwest Brazil. However, we're still consistently finding with new analyses that these three groups remain. I'm particularly interested in dealing with these major groups because for me, being a quantitative ecologist, what's really key is numbers, so the most localities. So there's not a lot I can do at the moment if we're just looking at one or two localities. So for the most part, these three major assemblages cover different time periods and environments. So the oldest, the Avalon, has fossils mainly found in Newfoundland in Canada and Charnwood Forest in the UK. And these are exclusively in a deep water setting, so well over a kilometre, around a kilometre deep, consisting, and then we move forwards to the White Sea assemblage, which is a bit younger, which is mainly found in Australia and in Russia, while the Nama assemblage is mostly found in Namibia. My work's mainly focused on the Avalon assemblage, but I'll also be talking about my work in the White Sea and indeed throughout the whole period. So we're starting off focusing on the Avalon assemblage. And so this is where we see widespread animals which lived in deep water, well below the photic zone, first appear. So most of the oldest Ediacaran organisms from the Avalon assemblage are taxa such as Charnia, which belong to this clade of organisms called rangimorphs. And they exhibit fractal style branching. That is, they have branches that have branches that have branches, sometimes down to 5 different levels. Now, these rangumoles are quite problematic. While they look superficially like plants, because they're found in deep water rocks, we know they can't have been synthetic. And their anatomies are pretty unique, with a fractal-like branching that's really only found in the Ediacaran, which means it's quite hard to use morphological approaches to work out what they were. So how can we go about investigating them? So what we do have is absolutely exceptional preservation. So here we've got Mistaken Point. This is the E and the D surface, and these are UNESCO World Heritage Sites because of their importance to understanding the history of life on Earth. And we can see here a very typical frond, got the holdfast disc that attaches to the seafloor, and then the stem, and then the frondose bit, which is upright in the water column. So these fossils are preserved as casts and molds on the surface in their thousands. And these immobile organisms were preserved under volcanic ash flows. So rather like Pompeii, everything was captured where it was living, creating multiple senses of Ediacaran life. So because of the exceptional preservation, it means that if we can map out these communities, we have a really exceptional data set available to us. But how do we go about creating these maps? So often in talks, we have these beautiful blue skies, but as anyone who's been to Newfoundland will know, it's not always like that. So this is a lot more of a typical Newfoundland day. And this means that while when the light's good, you can see these really exceptional details, it's a lot harder most of the time. And indeed, even when the light's good, we only get half an hour a day. The worst field season we had 2 half-hour slots of good light across the whole 3 weeks. So this, coupled to the accuracy of doing photogrammetry over very large scales, means that a different approach is needed. So what we've been doing since 2016 is using a laser scanner to map out these fossils. So here we've got the end of it, you can see carrying it, and here's the Charnwood discus. And we pass the laser scanner over the fossil. Here you can see the blue line, which is the line of dots. So this doesn't damage or hurt the fossils in any way, which is obviously very important because they're protected and we wouldn't want to. And then what you see at the end is a really wonderful 3D surface scan of the fossil. So here it's close up. So these are 1 centimeter grids, and we can see the holdfast disc, the stem, the branches, and some of the secondary branches. So the vast majority of the scans we've been doing are at a 40 micron resolution, but the new scanner that we tried last summer is down to 15 microns, so you get an amazing amount of detail. But of course, all this takes a rather long time. So here we've got a time lapse of me and Charlotte Kenchington scanning on the E surface at Mistaken Point. So this is about an hour's worth of work in 10 seconds, and we cover about 1 meter square. So you've got the laser which goes over the surface, and then you've got the mechanical arm, which helps get its accuracy, and of course a tripod, which of course has rubber feet at the bottom to make sure we protect the surface. And so here are some of the surfaces that we've mapped out and analysed so far. So here we've got the grey area at the areas that are mapped out. Each dot represents a specimen, the different colours are different species, and the size of the circle represents the height into the water column. And you can see you have a wonderfully wide variety, a huge beta diversity. So you have some surfaces that are very diverse, like E surface and bed B in Charnwood Forest, and others like H14 are dominated by just 1 taxa. You also have a large range of size. So Spaniards Bay is well known for its very small fronds, but then somewhere like bed B, you have really large fronds, sometimes 60 centimeters long. So what can we do with this? So these are the questions I'm going to be talking to you about today. So we want to know what's driving Ediacaran evolution. So we're going to use our data to investigate the role that competition has in the dynamics of these communities, what reproduction effects are, and how the environment plays a role as we go through into the outside the Avalon and into the White Sea and Nama. So because we have these wonderful snapshots of Ediacaran life, it means we can use these quantitative ecological approaches, which are normally only used in the modern, and apply them back to the Ediacaran communities. So of particular use has been spatial point process analysis to me, which has been developed primarily in forest ecology. Although over the last 5 years or so, you're seeing an increase in the amount of animal communities, so things like corals and sponges that are being analysed. The basic idea is you have your sessile organisms, you mark up the points where they all are, the different dots correspond to different specimens, different colors, different species, and different sorts of point patterns, such as these, correspond to different sorts of underlying processes. So for example, with this deep sea coral community, we can see they're very spaced out. And the reason they're spaced out or segregated is because it's very food limited. So they need a certain amount of water volume around them in order to survive. If two are too close together, there's not enough food for both of them. So one of them dies, and this leads to this thinning or spatial segregation. So key to these spatial analyses is the grouping and the hypothesis testing of different sets of processes into biological, reproductive, or neutral models, or niche or environmental models. So for example, if we have Thomas cluster models, this represents a reproductive process. So we have our parent and then we have the offspring around it. And the cluster is more dense in the center than to the side. Whereas if we have an environmental model, it's something more akin to if we're mapping out the trees in the ocean. So as we know, trees live on land. So if we're mapping out where the trees are in ocean, they correspond very, very strongly to where the islands are. And the primary signal we're going to pick up is that of the islands rather than the tree. So this is why they're environmental models. And so each of these different patterns has different sorts of observed processes. So by comparing our observed patterns, the red lines, to different sorts of well-known and well-established ecological models, we can determine whether we've got more reproductive processes, for example, or more environmental processes. So to start with, I'm going to consider what the utility of large body size or height was for the Ediacaran organisms of the Avalon. So Mark Clapham and Guy Narbonne suggested in 2002 that the main advantage of being tall for Ediacaran organisms was to avoid competition for resources from the water column. The idea was that different species lived at different tiers or strata of the water column. So rather like a rainforest with its shrub layer, understory and canopy layers, you have a niche which minimizes competition. So this meant that Ediacaran taxa were tiered in the water column to avoid competing with each other. So how do we go about testing this? So for this data analysis, me and Charlotte used the three communities that Mark and Guy worked on in the original study. So this is the D and E surface at mistaken point and lower mistaken point. Then there's a few steps. First, we need to identify resource competition within these communities. Then we quantify the extent of tiering within the other populations, and finally test to see if there are any statistically significant links between the presence and strength of tiering and the presence of resource competition. So how do we go about detecting competition? Well, spatial segregation results from competition, but it's not the only process that can do this. It can also form when two taxa inhabit patches which themselves are segregated. The spatial segregation is due due to the underlying habitat, not the interactions of the organisms themselves. And so in order to determine this, we start to look at our spatial point process analyses. So for example, we have our small scale segregation, which would look something like this. We have our habitat segregation and our resource competition segregation. But what's really key is thinking about how the different size classes behave. So when you have habitat segregation, you tend to have the small and large ones behaving in the same way spatially. Whereas if you have resource competition, the small ones, because they need less resources, are less likely to be segregated or weaker segregated, if they are at all, compared to the larger specimens. So that would look something like this. So you've got your small specimens that are coexisting very happily. You then, as they grow, they're needing more resources. And so the large specimens need a huge amount of resources, so we'll spatially segregate. Now, of course, because we have reproduction occurring not as a once-off event, it's actually all mixed together, so it's very hard to spot, which is why we need these techniques. So here we've got our first pair correlation plots, and so we've got the instances where we find spatial segregation with this differential size class. And there's only 4 instances that we found so far in the 8 communities that have been studied. And this is very unusual because on modern seafloor communities, everything pretty much competes with everything else. So in the deep sea, it's for food from the water. In the shallows, it's for space in coral reefs. So given the amount of abundant taxa that we can run these analyses on, we'd expect the equivalent to be 26 instances of competition, but we've only got 4. So they're not competing much. But then maybe the reason they're not competing much is because they're tiered. So we need to consider how much they are tiered. So here we've got a simple diagram. So we've got the heights and we've got the frequency, so the height distributions. So this would be an example of a fully tiered system because the size distributions don't overlap. This would be a very tiered but not totally tiered community, and where you see a lot of overlap between the size populations, then that would describe something that wasn't very tiered at all. So what did we find? Well, here are the 4 communities we've looked at. So we've got B surface on Charnwood, but we've got D, E, and mistaken points. So we've got the species along the x-axis and the height along the y-axis. So where it's very dark, you have more of specimens that occupy that particular tier of the water column. And so what we can see on D surface, it's very tiered. You've got this diagonal element. However, that's not what you see on the other communities. You can see they're very untiered. 7.9% on bed B, and E and low mistaken point are very, very overlapping. So we're not finding tiering. So what's going on? So one approach would be to break down the multiple abundant populations we have in E surface and do pairwise tiering metrics to see what's going on directly. So Fractifusus and Primo Candelabrum are very tiered, and they also compete with each other. Primo Candelabrum are about the middle. However, Biofucus and Braggatia, despite their low metrics, don't compete. And indeed, when you run the stats, you find there's no correlation between the presence or any the strength of competition with any of these tiering metrics. But all's not quite lost, because we still know that D is very different. So we know that D surface has no instances of competition on it, and it also is very tiered. So maybe we can investigate what's going on with that. So what we did is we looked at the community compositions of these 3 different surfaces and broke them down into 2 different groupings. The first one was to consider rangimorphs versus non-rangimorphs, because D is very much dominated by the rangimorphs. And the second one was to look at stemmed versus non-stemmed taxa. So we didn't find any significant differences between the rangimorphs and non-rangimorphs, but we did find significant differences between the stemmed and non-stemmed groups. But it wasn't what we were expecting. So we thought that stems evolved in order to ensure species reach new tiers and were higher in the water column. And if that was true, we'd expect stemmed organisms to be very tiered in contrast to the non-stemmed ones. But what we found was the opposite. Stemmed organisms were very, very overlapping, whereas the non-stemmed ones were highly tiered. So this then raises the question, what are stems good for? So one possible option is that they're helping to maintain, helping to survive in turbulent conditions. So this was a paper done by Phil that came out a couple of years ago, looking at the orientations on the E surface and hypothesizing that the organism, or showing that the organisms without stems tended to have more variable orientations than the ones with, suggesting that maybe they were less robust to incoming turbidite flow. The other possibility is, because we spent a huge amount of time looking to find out what the advantage of being taller was, and the only advantage we could find was that taller fronds have larger dispersal clusters, so that they've advantage in terms of reproductive competitiveness. The taller you are, the further your propagules, your offspring can go. So we haven't been able to find any evidence that vertical competition is really driving the ecosystem dynamics. But what's going on horizontally? So we go back to our spatial models, and we're thinking about in terms of neutral biological models versus neutral environmental models and how that changes across the different surfaces. So for this study, we had 7 surfaces from Newfoundland, Canada and Charnwood Forest. This includes over 10,000 specimens and includes the vast majority of all species known at this time. So what we did is we fitted a whole bunch of different models, combinations of reproductive, niche models, random models, and mixtures of niche and reproductive models to test what the best fit models were for this data. And what we found is there's a massive, massive weighting towards neutral models with very few instances of niche models. And what's rather interesting is you get the same sort of patterns occurring across multiple different surfaces. So this is suggesting very much that we're not having strong environmental drivers very early on, in animal life, in the Avalon, and suggesting that perhaps any diversification was a lot more stochastic and due to maybe differences within the population rather than a systematic adaptation to the environment. But what happens as these communities develop? So for his PhD, Niall Stevenson is investigating how communities develop through time, i.e. what's the nature of community succession in the Ediacaran. So if we think about our kind of modern day community successions, we have pioneer plants that come in and then grasses grow up. We've got shrubs and then short-lived pioneer trees before we get to our climax forest. And this is the communities are developing in very predictable ways. So we can assess what's going on with community development by testing patterns of relative abundance versus relative biomass or sizes and seeing how they compare in order to quantify the stage along community succession. Then we can compare the relative successional stage back to what the community composition is. So the suggestion made by Matt Clapham again was that we start with reclining fronds, so things like fractifusus, and then over time we progress to taller and taller fronds through different interactions such as pioneer species, making the seafloor more habitable, but then being outcompeted. So demonstrating what the better competitors were and then what's driving evolutionary dynamics. So this would look something like this. We've got our early communities with our pioneer species, and then these slowly change and decrease in abundance as you get your climax species coming in. So what did Niall find? So these are plots of the relative abundance to biomass. So when you have lots of little ones, what that means is you have a lot more abundance to relative biomass, and that changes as the succession progresses. So by quantifying this, we can determine that we have our early successions, and as we go through, we start to get more middle succession communities, until we end up with our late community developments. And so this is why, in this way, Niall's managed to quantify these 3 different stages and put the different Ediacaran communities that we've been studying in these different groups. But what you find is not what you'd expect. So we've got our early communities here, our middle ones, and our late. And as should be quite clear, there's a huge range of variation with the community compositions, and there isn't a systematic changes and consistent changes of community composition as the communities develop and mature. So if we're talking about competition, I've shared a few ways that we can investigate the different sorts of competition, finding that resource competition is actually rather limited within the Avalon. Height doesn't provide a refuge from competition, and indeed tiering is relatively limited. So height maximize dispersal, offspring dispersal, and then stems possibly helped with survival. We've also shown how community development, it seems to be very stochastic and random. It's not systematic in the way that you might expect from the modern. And this all comes together to suggest that Avalonian dynamics are really rather stochastic, much more about random chance differences within the population, who colonizes who at what point, than about a systematic adaptation to the environment. But what's going on with reproduction? How does this feed into the ecological and evolutionary dynamics? We know very much that the key parts of natural selection are organisms reproducing, survival of the fittest, and adapting to their local environment. So if we, for this study, this was the first of the spatial studies, we looked at fractifusus in Newfoundland. This is pretty much endemic to Newfoundland. There's one specimen, possible specimen that's been found in the Mackenzies. But the reason we focus very much on this is it's really super abundant. So for this study, we used 3 communities, each with 1,000 specimens. I think between us, me and my group, we've now mapped out probably closer to 10,000 fractifusus. There's a lot of them, which is fantastic if you like your quantitative analyses. So fractifusus live directly on the sea floor. And you can see some here from the Discovery Geopark in Buena Vista Peninsula. So going back to our models, what we're really zoning in on is these reproductive patterns. So finding them, establishing that's what's dominant, and then seeing if we can work out what's going on with their reproduction. So what did we find? So on D surface, we found a single Thomas cluster. So what this is telling us is we've got a single reproductive event. Here's the radius here, and this radius here corresponds to the radius of the clusters. on E surface, we had a double Thomas cluster. So we have our initial reproductive event followed by a secondary reproductive event. So we have clusters of clusters, and we have the same pattern that we find on H14. So this indicates reproductive events because we always like to check ourselves. We then looked at the size-based spatial distributions, showing that the large ones are randomly distributed, the medium ones form a single Thomas cluster, and the smallest double Thomas cluster. So that, and furthermore, the small ones cluster around the medium ones and the medium ones around the large ones. So these size analyses confirm multiple reproductive events. So what's going on? So we want to know what the dominant mode of reproduction is on these surfaces for these organisms. So in the modern benthic organisms, we have three main modes of reproduction. We have fragmentation or budding, we have the release of waterborne propagules, such as coral spawning, and we have reproduction via stolon or runners, like spider plants or strawberry plants. So in the 10,000 odd specimens we've mapped out, we do see some fragments and we do see some smaller fractifuses that look like the chunks, but the vast majority of them, like the small ones, look like small versions of the bigger ones. And so there's no way that this fragmenting or budding can be the dominant driver for reproduction. So the question then becomes, is it by waterborne propagules or via stolon? So we can look at that by looking at the directionality of the clusters. So not the individual specimens, but the clusters, the aggregation clusters that they're producing. Because if you're releasing propagules into the water column, they're far more likely to go directly in front of you with the current than they are either side. Whereas if you are maintaining links to your parent via stolon or runners, you're less subjected to the currents, which means you form rounder clusters. And this can be seen using these density plots. So here we can see this is random, the blue, and we can see here for the largest ones, they're very, very directional. We have 4 times as dense in one direction on either side. But this isn't true for the medium and the small ones, where we can see, note the different axes. Actually, the variations 10 to 20%. They're a lot more round. So what this means, we think, is going on is you have an initial colonization event where fractifuses are colonizing from the water column onto a new substrate. They then reproduce quickly, asexually, using runners or stolon, and they reproduce again. And so we have these dual mode of reproduction. So what's really exciting is about 5 years after this paper was published, some colleagues, Alex Liu and Frankie Dunn, found 5 different Ediacaran taxa, including fractifuses that had evidence of stolon. So here's a photograph from the Matthews surface, LC6, in Little Catalina in the Discovery Geopark. You can see the filaments coming in, attached to the ranking-morph again and again. And if it's hard to see that, we've got an overlay of this. So we've got these connected filaments connecting the different ranking-morphs. Further evidence for fossil stolon has been provided by Katie Dallhook. Now, she's been working on casts that we made back in 2018 on H5. Here's a cast, and we've got Katie on the H5 surface. And she found something really, really exciting. So she found what she termed conga lines. So these are small fronds, often quite effaced, and sometimes ending with a large frond. So the first question is, what is the chance that this happens just randomly? If you spend your life looking at thousands upon thousands of fossils, sooner or later they're going to appear to line up. Humans are great at pattern matching. So what's the chance these could have occurred just by random? And Katie worked through all the probability models. And so in this case, a chance that you get the number of conga lines that you'd expect on H5 just by chance is less than 0.01%. So we know they're a real thing. They're not just artifacts of our hopeful imaginations. So in terms of what processes could cause conga lines, if we focus just on sessile organisms, the key options are stoloniferous reproduction or fluid dynamics. So Katie then developed models using CFD and particle tracking to determine from multiples of different current velocities what the length of the conga lines would happen if you have this fluid flow over a frond here, it gets kind of whipped back round, and if the kind of propagules are settling here, what's the chance that this creates this conga line? So we can see here her results. This is the length of the conga line in centimeters, and this is the predictions made in different velocities. And you can see the observed conga lines are much, much smaller in length than what you predicted. So this, she's concluded that stoloniferous reproduction is the only way that you could get this effect, which is wonderful because it's another different sort of stoloniferous reproduction that we're finding in the Avalon. So these stolon and competition results, rarity of composition, motivated the current research fellowship that I'm working on, which is testing the hypothesis that if asexual reproduction by stolon dominates, and if connected individuals share resources, as is the case with animals and a lot of plants, via these stolon, then you have the potential to reduce competition between individuals, because there's no need to compete with your neighbor if you're sharing the same resources. And this then could mean that stoloniferous reproduction explains why the Ediacaran competition for resources is relatively rare and weak. So how do we go about testing this idea? So we need to detect it. So we're looking for extreme dispersal limitation, and we're also looking for the directionality of the clusters. So very, very long and thin ovals versus circles. We detect competition using spatial segregation, and then look to see if there's a correlation between the two. So here we've got some examples. We've got our reproductive models, and this is randomly simulated. And you can see the relatively round directionality of the clusters. If you then force the directionality on it, like you'd expect with currents, you then have a lot more variation. If we compare to modern living things, this is a hydroid colony with stolon, you can see that it's relatively round, especially when we compare to modern communities. So this is a wonderful sponge and coral community in the Pacific Ocean. Some of these sponges are 5 or 6 meters high. What's really exciting is if you look at directionality, they're very current influenced. And what's more, there's two parts to this. There's a kind of the slow kind of, I guess, gradient up to the ridge and then a sharp drop off, which has a different current. And you see that reflected in the distributions of the sponges. So we've got these two approaches. So what did you first find? So we look for intra-specific competition, that's competition within each community. So it's detected by a size-based spatial segregation. And the thing to bear in mind with the relative strength can be assessed. So if you've got a PCF of say 0.7, that means you have a 30% reduction in your, or thinning out of your community. So the 20 taxa populations analysed, 5 had these sorts of size-based structuring that indicates resource competition. So the first question is how does that correspond to the directionality and the kind of the factors associated with stoloniferous reproduction? So here we've got the directionality of various different populations across different surfaces. You can see some are very highly directional, whereas others aren't. And what we find using a binomial regression is there's a significant difference between in terms of directionality variation between the populations that have significant spatial segregation and the ones that don't. Furthermore, if we're looking deeper into the strength, we can see here we've got what's actually observed versus the predicted segregation based on the stoloniferous factors, so the size of the dispersal clusters and the directionality. And it's got a really, really good model fit. We're talking about 0.51. So that's over half of the variation in terms of spatial patterns and competition that's predicted by factors relating to stoloniferous reproduction. So where it gets really interesting is where we start considering how the intra and interspecific competition relate to each other. Normally, in the modern, you'd expect intraspecific competition, so that's competition within the population, to occur at much smaller spatial scales than between different taxa, because reproductive processes and habitat filtering mean you're far more likely to be close to the same species, but also have the same requirements, so you're much more likely to compete with a neighbor of the same species than you are with a different one. However, you can sometimes get this not occurring, where you get competition between species occurring at smaller spatial scales than within a species, and this is not called heteromyopia. And heteromyopia happens when the same species has a big advantage to being close. So it could be because the kind of parent releases chemicals to mediate pathogens or predators, for example, or it could be because you have interconnected clones or gametes. So I think we can all agree that we're going to exclude predators as a source of Avalonian heteromyopia, because there's absolutely no evidence of them whatsoever for at least 10 million years. Pathogens are also going to be excluded because they act strongest at lower radii than at larger ones. You'd expect the highest density to be where they're, essentially R equals 0. And with the exception of bed B, we don't see this pattern. We actually see higher amounts of aggregation at small spatial scales. So what this tells us is we have further evidence of stoloniferous reproduction across some, but of course not all, taxa. And what this is telling us is that because we have this stoloniferous reproduction and they're remaining connected, these communities, the organisms, were functioning very much as teams rather than individuals. And what's really interesting is this heteromyopia is a stabilizing mechanism, which can increase diversity because the extreme aggregation and dispersal limitation that occurs with this means there are often gaps that weak or rare competitors can occupy, so that one or two species don't necessarily dominate. But it's relatively rare today, which suggests that perhaps the Ediacaran systems were occupying a very different sort of regime. However, we know from modern literature on plants specifically that if stolon are broken, and this could happen by burying or predation, or with some experimental studies, researchers cutting the stolons between all the different gametes, then the system destabilizes and different stabilizing mechanisms are needed. So this could potentially explain what's going on with diversity dynamics later on in the Ediacaran, where as you get the evolution of burrowing, it's changing the system dynamics. So to wrap up what we know about Ediacaran reproduction, well, we've demonstrated how we can use spatial analyses to resolve pretty complex dual mode reproductive traits, and that often we get asexually dominated clonal reproduction. We've also been finding multiple different modes of stoloniferous reproduction and showing that actually competition is very much dependent on reproductive mode. So the final thing I want to talk about is the effect of environment. I hope you will all agree the environment has very little impact for the Avalon, but what happens when we move forward in time? So for this study, we're going to expand out our temporal and spatial scales to include younger surfaces from Russia and Australia. So as we move into the White Sea from the Avalon, we see a large increase in species richness, about tenfold. With this jump in taxonomic diversity, we also see a jump in morphological diversity. We get many bilaterian species, such as Aspergina and Dickinsonia in their thousands, as well as Phanerozoic, which Mary Drozer and her colleagues demonstrated was reproducing slightly sexually via spat form. It's also found preserved either as its holdfasts or shown here, or frill body fossils. Now we need to be a little bit careful because when we start getting mobile organisms such as Cambrian and Dickinsonia, it's much harder to fit that to known mobile patterns, and so we're describing the patterns that we find a lot more than the we don't have that bank of models that's available for subtle organisms, so it's hard to analytically determine what the driver mechanisms are. So what did we find? So here we've got a summary of all the results from 14 different communities from the Avalon and the White Sea. And what's very, very clear is there's a significant difference between the key processes that operate in deep water versus shallow water, with the deep water exhibiting much more biological or neutral processes and the shallow water very much niche processes. So while there is a temporal difference, unfortunately, we haven't managed to get, well, it's not possible to get accurate enough dates to really analytically test whether this is a temporal trend or an environmental trend. What this is showing us is that in the shallow water, the driving factor behind the population dynamics is the organisms interacting with local habitat patterns, local habitat heterogeneities. And we have a significant correlation between the species richness and the extent of interactions with the local habitat. So our results are particularly interesting when taken into account with the previous hypothesis of early animal evolution. So Graham Budden, Soren Jensen introduced this savanna hypothesis to explain early animal diversification. whereby early bilaterian diversification was driven by small-scale variations in the local habitat, primarily caused by the spatial distributions of settled taxa. So they argued it's a drive to find these heterogeneous resources that led to novel evolutionary innovations such as mobility and eyes. And in a similar way, the early human evolution is thought to be driven for searching for resources in the African savanna. So our results very much demonstrate that at least some of these early animal communities were indeed influenced by these habitat patching variations, and that changes. It's not a given from the start, it's something that develops. And we did very much describe a mechanism that links this early animal diversification and benthic habitat patchiness prior to the evolution of predators and widespread pelagic organisms. So how might this adaptation to heterogeneity be taking place? So to consider this, we looked at some examples of two trace fossil assemblages, one from the Ediacaran, the Shibatan, and also from the Cambrian of India, Nagoa. And so what we did is we looked at we marked up where all the trace fossils were, and looked at not where they were, the lengths and the angles between the different movement segments. So how might this work? So if we have a patchy environment with an animal that doesn't care about it, that's not interacting with it, it's going to be moving in a random walk. However, if it's capable of sensing the environment, then when it gets to a good quality patch, it'll spend more time. And this means that the kind of trace lengths will shorten and the angles between them will change. And we can test for this using marked correlation functions, which is looking for spatial autocorrelation between the values of the trace lengths and the angles coupled to where they are. And what we find is different when it comes to the angles between the Shibatan and the Cambrian Nagar. What we find is that in Shibatan, the traces don't have this shortening of angles, so it's the kind of turning around when they get to a good optimal patch, whereas you do see that happening in the Cambrian. So while this is only 2 samples, it's very suggestive that you've got this change of happening. So this is a kind of a mechanism or a possibility of one of the many ways that these animals are adapting. So the final thing I want to talk about is looking through the entirety of the macrofossil assemblage, focusing on the main ones. As I said, mainly because it's a data problem. We don't have the detailed data to look at what's going on outside these 3. And what we're looking for is a structure within each of these assemblages. So as we move through the Ediacaran, we can see this uptick to the White Sea, but we also see this big diversity drop, which has been suggested to correspond to a catastrophic mass extinction. So what we're looking for is looking at how ecosystem dynamics change over this period and how they fit into our expectations about mass extinctions. So there have been a lot of different studies over the years looking at this diversity dip, and they very consistently find that it couldn't have been an artifact of sampling. And this is where you vary kind of how you define taxa and how you define different things versus number of sites, suggesting that this diversity dip isn't sampling bias. However, I wanted to just draw your attention to the supplementary info of a recent study done by Kat that demonstrates this isn't necessarily true across the board. So Kat's study looked into the differences in patterns of different sorts of Ediacaran groups between high and low latitudes. She found some really interesting patterns, such as a low to high radiation of bilaterians around 550 million years, but also a high to low radiation of frondomores post-gaseous glaciation. But the result I wanted to point out here was that in terms of sampling biases, this varies too, with the low latitudes showing no significant difference in terms of taxa with number of localities, i.e. there is a sampling bias. And as such, we need to be a little bit cautious about dismissing the sampling artifacts out of hand and really try and think about where and how they might be affecting our interpretations. So the study then that I'm going to talk to you about is looking at these diversity dynamics and what could be happening from within the assemblages. So for this data set, we used the data that was compiled by Bogatal in 2016 and then used more by Muscanti in 2019. So here we have all our data. It's been ordered by taxa with similar community compositions, which are close together. We can see the Avalon, the White Sea, and the Nama. You've got the localities here on this axis, the relative depth, and the taxa here. with different groupings of different sorts of taxa. So in all, there's 86 different localities and 124 different taxa, and they're represented by the Avalon by 30 sites, White Sea 29, and 12 by the NAMA. So for this study, we use the elements of metacommunity analyses. So this is a way to quantify structure within the metacommunities. So if we think about Berg's results, it's within the NMDS clusters, trying to work out what's going on between, and not between the assemblages, but within them. So it's within each assemblage, what is the structure between the different communities? And so we have similar sorts of patterns. We have random, so this is taxa versus localities, presence, absence. If they're there, it could be random pattern, we could be segregated. This is also sometimes known as checkerboard scores in modern ecology, or we could be aggregated or clustered together. And things start to get really interesting when you look at aggregation. So if it's checkerboard, there's not a lot you can do, but there's a lot you can do if you have aggregation, because you can then split the aggregation, the coherence, and look at two different metrics: the turnover, which is the amount of replacement of species between sites, and the boundary clumping, so the overlap of species ranges relative to each other. So if we take, for example, what happens when we get a negative turnover, we have a nested species lost, is what it's referred to. So communities are subsets of the total population. And this corresponds to wide environmental tolerances with little specialization. And you use these boundary clumping metrics to determine the degree that species are grouping together. Alternatively, if you have positive turnover, what you're seeing is a community that's specific to the background environmental gradients. And how the species respond to this environmental gradient can be determined using the boundary clumping to whether they do it individually. So it's individuals interacting with their environment, or whether it's a community-wide thing. So there's potentially synchronous responses to the environmental gradients and trade-offs between different taxas through life history traits through time. So what do we find? So for the Avalon assemblage, we see significant positive coherence and boundary clumping, but we see significant negative turnover, which is characteristic of a clumped species lot. So very, very little significantly non-random relationships. So besides metacommunity analysis, sorry, I should say, our student Becky also ran some co-occurrence analyses. So these plots here, they're where taxa occur together significantly more than you'd expect by random. So there's not just where they occur, it's whether they're more than you'd expect by random. And you can see very limited amount of significant pairwise co-occurrences. The UK sites do form subsets of the Canadian sites. So when we move to the White Sea assemblage, we see very different. We see a highly structured response. We also see differences between the Russian and Australian sites. And what's really interesting is where you see the same pairs of taxa occurring in Russia as well as Australia, they behave differently. Sometimes they're positively correlated, sometimes they're not. But you're still not seeing any significant correlation with depth. When we move through to the NAMA, we're seeing an even more highly structured system. So we're seeing biomineralizers found in shallow waters with soft-bodied fauna in deeper water. And this isn't a particularly new result. You know, people already were very much aware of this pattern, but this is a statistical kind of analysis demonstrating what, to be fair, a lot of people who were lucky enough to work in Namibia already knew. And we see this is a significant correlation with depth. So if we take these all together, what we're seeing is increased specialization through time. We start off in the Avalon with limited specialization, limited significant correlations between taxa and very wide environmental tolerances. We then, as we move to the White Sea, we have synchronized community-wide response to the environmental gradients. Before moving through to the NAMA, we see significant environmental segregation and depth structure. So what does this mean in terms of the diversity dynamics? Well, it suggests that actually we're not getting a catastrophic mass extinction occurring here, because if you have mass extinctions, you tend to have a lot more generalists occurring rather than more specialists, and we see more specialists in NAMA. You also see a maintenance or increase in significant pairwise taxa associations, which again suggests a long evolutionary relationship that's continuing. and you see positive turnover. And these are all things that are not associated with catastrophic mass extinctions. So potentially what we're seeing instead is an internal restructuring of the communities through specializations. So to summarize the work we've been doing, in the Ediacaran, we see a stochastic start to animal life, with random chance potentially playing a much bigger role in the evolutionary dynamics than systematic adaptation to the environment. We see that reproductive mode has a really strong effect on the dynamics, especially in reference to the community composition, potentially also driving diversity dynamics. We then see that as we expand and get younger, there's a drive to find these heterogeneously distributed resources that potentially led to novel innovations such as bilaterians and mobility, before finally ending up, just for Cambrian, with really rather highly complex ecological systems and a lot of niche specializations. So I'd just finally like to thank all my wonderful collaborators. Of course, none of this work can be done in isolation. We rely very heavily on lots of wonderful people to work with, and of course, my funding bodies. So thank you very much for listening.
Thank you a lot, Emily. That was great. We're going to transition to the question and discussion section, and you can ask by either raising your hand virtually by finding the raise hand button, or you can type it and I can ask it for you. First, Emily, I noticed that my reminder email to everybody last night never sent, so I'm very sorry about that.
Okay.
But I have several questions that were sent as direct message to me in the chat from Karthik Sender Sarwan, so I can start with some of those. He first asked early on, are isolated holdfasts or isolated from their fronds counted as unique individuals or not taken in a consensus?
So when we're looking at isolated holdfasts, we map them out, so one of the analyses I did with Nick Butterfield was looking at how kind of these holdfast discs related to the different frondos where you see the entire specimen, and at least on the surface, they definitely form subsets of what you'd expect from the frondos organisms with holdfast. But for the majority of when we're looking at these analyses and doing them, we're looking at the abundant taxa, and that's the full frond without the discs, even though we've done sensitivity analyses to kind of investigate this.
Okay. They also ask, does the net arrangement of all instances of organisms in a particular bed have anything to do with the flow of water or water movement around the organisms, and would this result would the resultant effect have strong ramifications on their survival as filter feeders? If yes, does this indicate mutualism between these bendobionts?
So this is a really interesting question. And so Imran Rahman and Simon Darroch have got an ongoing NSF NERC project looking into the computational fluid dynamics of communities, which I've been involved with, doing the simulations of the spatial patterns of these communities. And what's really interesting about that is that you can test these hypotheses, right? So you can see how the fluid flows over different organisms, and then you can see, using things like the co-occur analysis, how this plays out across multiple different surfaces, and through the spatial analysis. If you've got strong mutualisms, you'd expect to find that spatially, and you'd expect to find that across multiple different surfaces, and see how that then ties in to the results of the fluid flow. And as far as I can tell, we're not actually seeing any significant impacts for the work we've done so far on between the fluid flow and how a fluid flows over these organisms with the kind of broader scale ecological patterns.
All right, the next one they have is, are smaller or younger individuals of a species clustered in certain regions of the beds, hinting to a presence of a sort of basal nursery?
Sorry, could you repeat that? The sound wasn't great.
Sorry. Okay. Are younger or smaller individuals of a species confined to certain regions of the bed, hinting at a presence of a sort of basal nursery?
Not in the Avalon. I wouldn't want to suggest what's going on in Nilpena, for example, or in the White Sea. But you don't see that at all in the Avalon communities.
Okay, then what the next one, would applying optimum foraging models in the White Sea assemblage lead to the results on the development of locomotion?
So in the trace fossil study that I mentioned, so this was done with Scott Evans, who's now in Florida, Xiu Hai and Shi Zhen in Nanjing. I did a huge amount of analysis to look at the spatial patterns and try to model it as different sorts of optimal foraging patterns. And basically there wasn't anything interesting. They behaved very, very similar, not very optimally, to be honest. So I think this is something where I think if you had a lot more data over a lot more time, you could potentially start teasing out these patterns. But you don't see that yet, or at least with the two samples. This was a lockdown project. we unfortunately weren't in the field collecting data.
Okay, I jumped over a couple here, I just noticed. Okay, does mapping of these communities point to any taxon being a keystone species in its environment?
So I wouldn't say mapping does, but one of the sets of analyses that we've been using is Bayesian network analysis. And so what that does is you can look at you can use it spatially or temporally, but what you're doing is you're looking to see which is the most connected species to the other taxa. And in the Ediacaran, you're not seeing these kind of keystone taxa. Work I've done with colleagues in Antarctica, you see very strong keystone species, particularly sponges. And sponges have a huge impact on the environment beyond just being a habitat for other organisms. So you can detect this, but we're not seeing that in the Avalon at least.
Okay, and there's one more, and I'm going to send it to you because I don't want to butcher this word. You may have said it, but I may have missed it. Did you get it in the chat?
Sorry, I hid the thing.
Oh, you can't see the chat here.
Let's.
It's okay. I'll try to say the word. Okay, do the positions and composition of Ivesheadiamorphs, okay, contribute any information about competition?
So they don't to competition, but what's really cool is, so Alex came up with this hypothesis. Sorry, excuse me. That Ivesheadiamorphs are the decaying remains of other taxa. And so in the paper I did with Nick, using Bayesian network analysis and the spatial analyses, you can test to see whether the Ivesheadiamorph spatial patterns are what you'd expect if other organisms were dead and decayed. And what you find is really cool. So you find that the Ivesheadiamorphs have the same spatial patterns that you'd expect if Fractifusus and Chionodiscus were dying and leaving behind the Ivesheadiamorph spatial pattern. But the other cool thing is lobate discs, who are kind of sort of taphomorph potentially, were suggested, I think, to be the kind of taphomorph of a holdfast disc, but but they behave very, very differently to all the different fronts that suggest that actually they're not doing that. So while Ivesheadiamorphs don't contribute directly, so I've got a tickle in my throat. Of course, you start getting paranoid about coughing. You can kind of detect these sorts of patterns. So I think there's lots of interesting work that can be done looking at Ivesheadiamorphs across different surfaces and seeing if you see the same patterns occurring.
Okay, well, huge thanks to Karthik for a roll of great questions there. I'll leave it open to anybody else who wants to go ahead and ask. If you have anything you'd like to say, please go ahead and speak up. Oh, we have a question from Heda Agich. Go ahead, Heda, please.
Hi, Emily. Thanks for a wonderful talk. So you mentioned that competition is based on reproduction. So I'm wondering also, is what you're seeing in these rocks, is reproduction also dependent on the environment in any way? And here I'm specifically thinking of, modern sessile organisms like ferns, for instance, who can sort of change their reproductive strategies based on changing environmental conditions. Is this something that you're seeing at all?
Well, I mean, so when we're looking at kind of dual modes of reproduction with fractifusus, you do see that the different things, and actually there's a vast majority of living things can reproduce both sexually and asexually, which is almost disappointing because it would be nice to be able to use this to restrain it a bit more. But in terms of patterns between surfaces, what is really, really interesting is you're seeing exactly the same reproductive patterns occurring across multiple surfaces. So, for example, with fractifusus and Charnwood are really notable ones. So fractifusus, the H14 surface, discovery surface in Discovery Geopark is, I mean, I think it's about 800 kilometers between them at the moment. Potentially, I'm not sure, they're not well enough data to know precisely how different they are, but there's a big gap and they have exactly the same spatial patterns, like statistically the same spatial patterns. And also, if you subset E surface just into 2 size clusters rather than 3, it has the same patterns as D. And you're seeing exactly the same spatial patterns occur, which indicates exactly the same spatial reproductive mode across many, many different taxa. And you're also seeing that with Charnwood. You see the same double cluster, Thomas cluster patterns occur on the E surface as you do on bed B. And that's, again, kind of, huge amounts of time and distance potentially that separates them, and it's exactly the same.
Thanks.
Okay, Andre, you unmuted yourself there. Did you want to ask a question?
Yeah, Emily, great talk. I was wondering, so Avalon is the oldest assemblage and it's already deep water. And I'm wondering what thoughts you have in terms of being pushed into deeper water environment by sort of competition, and since its earliest setting, sort of the competition in deeper water environment did not establish yet at that time.
Sorry, the sound isn't great, or my hearing, or both of them. So was that question about the kind of competition and what the competition is for in deep water, or?
So if you have any thoughts in terms of Avalon being the oldest assemblage and yet deeper water, if it was pushed into deeper water environments by competition in shallow water environments.
Oh, I see. You're suggesting a kind of shallow water to deep water migration.
Yeah, and if that's the case, when competition in deeper water environments was not yet established because we just migrated there.
Yeah, I mean, so yeah, in theory that could be possible. I think one of the really interesting results that Kat found when she looked at the paleolatitude differences is that you actually kind of post-gas you see a similar, you don't see any significant differences between distributions of fossils in the low and high latitudes for algae and for protists, but you do with frondomorphs. And then with frondomorphs, they start in the kind of deep water, high latitudes, and then radiate into the shallows. Of course, there may be shallow water organisms that we're not finding much of, apart from the landyan, potentially. But yes, this absolutely it could be feasible. It's always difficult where you don't know. There'll never be a time where we can draw a concrete line and say, okay, we've all looked for decades and no one could find substantial shallow water kind of post-Gasquet localities. So does that mean that they're not there? Like, where do you draw the line? But yeah, it is, yeah, of course, possible.
Thank you.
Okay, Greg Ritalik's hand is raised. Go ahead, Greg.
Hi, Emily. I'm trying to start the video here. There we go. That was really brilliant and very thought-provoking. I really enjoyed it. What you're doing is really pretty amazing. And I'm wondering, is there any possibility that any of your, I mean, you use a lot of analogies with plant succession and with terrestrial communities and all this sort of stuff. Is there any possibility that any of these analyses you do could actually discriminate between a marine and a non-marine community?
No, no. I think that's very much for my my geological colleagues to determine, because the patterns you're seeing are very, very variable and very different. But then they're different to everything. So there aren't consistent. Well, in the modern, there aren't consistent terrestrial patterns and marine patterns or freshwater patterns. They're all mixed up, and it depends on a multitude of different factors. So yeah, there isn't a strong modern pattern, so you can't use that to then try and infer what's going on.
What a bummer. Yeah. I still haven't lost hope, though. I think what you do could actually decide it. But well, I mean, I can't think of a way either. Yeah.
Well, so I mean, it's about what data you have. So I mean, I've been working with a lot of modern ecologists to try and help build up the kind of the database, if you will, of what sort of patterns occur in the modern and deep sea and in Antarctica. And this summer we're going to look at shallow water soft coral communities. But yeah, if they're all if they're very different to the Ediacaran, but then the marine and the I mean, if we think of like an MDS plot or something like that, if essentially the terrestrial and marine clouds perfectly overlap, and Ediacaran is potentially there for the Avalon, and then maybe coming closer to the White Sea, there's nothing we can do. If you're not finding the patterns in the modern, you're not going to be able to apply them back to the Ediacaran.
Is there a possibility in that last graph where you showed the Avalon separate from the others that you could plot some modern communities, both terrestrial and shallow and deep marine, on that plot?
Yeah, and the earlier versions of the talk I did. I mean, it's very, very interesting, because when you get to the White Sea, when you're in terms of metacommunity structure, in terms of co-occurrence, you're then getting to the same sort of metrics as you get in most of the modern, and the Avalon is very much an outlier. Right. There's a lot more modern work that's done on metacommunity analyses and co-occurrence analyses than with the spatial stuff.
Well, fascinating, and the rigor you're bringing to this is really impressive. I'm amazed. Thanks very much.
Thank you.
Okay. Anybody else? If you feel you have a question still, go ahead and feel free to speak up. I'll leave a few seconds for it. All right, well, thank you a whole bunch, Emily, for a really great talk and really great discussion. It was really great to have you, and this is a great contribution and a unique type of work that we don't see very often. Thank you a whole lot for your contribution here.
Thanks very much for inviting me. Very much enjoyed it.
All right, great. Well, everybody, stay safe and have a good weekend and see you next time.
Thank you, Emily.